Welcome to Symposium 2 on Model Sharing, Acceptance, and Communication with FDA. You'll be hearing from 4 presenters in this session. In order of presentations, our first speaker is Dr. Liang Zhao, who is the Director of the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD, CDER, FDA. Dr. Zhao has established his leadership in industrial R&D, quantitative methods and modeling, and model-based strategical decision-makings in regulatory and industrial settings for generic and new drugs. Our second speaker is Professor Andrew Hooker, who has been trained in physics, mathematics, bioengineering, and pharmacometrics at the University of Colorado, Washington, and Uppsala, Sweden. He is currently a professor of pharmacometrics at Uppsala University, Sweden, where his focus is on methodological pharmacometrics questions. The third speaker is Dr. Viera Lukacova, who is a chief scientist at Simulations Plus. Over the last decade, she has been contributing to the research in the area of magnetic absorption and PBPK modeling and the development of GastroPlus, DDD+, and MembranePlus software packages that has been widely used. Our fourth speaker is Professor Amin Rostami, who is a professor of system pharmacology and director of Center of Applied Pharmacokinetics Research at the University of Manchester. The scientific work of Professor Rostami covers wide areas of drug development over the last 30 years, ranging from pharmaceutics to clinical pharmacology and systems pharmacology. We are going to have 2 moderators for the live panel discussion. Our first moderator, Dr. Lucy Fang. Dr. Fang is the Deputy Director of the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD, CDER, FDA. Dr. Fang is an established expert at the FDA in the use of quantitative clinical pharmacology approaches in the review and regulation of generic drugs. Our second moderator is Dr. Mark Sell, who has more than 30 years' experience in population pharmacokinetics, pharmacodynamics modeling in both academia and industry. In his current role as Vice President at Cytara, he is responsible for conducting and overseeing population pharmacokinetics, pharmacodynamics analysis, and strategical consulting on drug development. Once the panel discussion goes live, the moderators will introduce the rest of the panelists joining in the panel. We hope you're enjoying the session. Now let's turn on our attention to the presentations.
Thanks for the introduction and good afternoon, everyone. My name is Liang Zhao, Director in the Division of Quantitative Method Modeling, or as OGD. Hope you have enjoyed all the sessions so far. Now it's my turn and my great pleasure to introduce the concept of model master file and its potential types. Disclaimer: this presentation reflects the view of the author and should not be construed to represent FDA's view or policies. We have been observing an increasing value generation of modeling simulation in drug development. As for bioequivalence assessment, quantitative method modeling are not only a technical tool set, but also the quantitative accurate summary of information and knowledge throughout a product life cycle. QMM provide the critical knowledge base for regulatory decision makings for all subject areas we have covered in the workshop. Here I want to briefly mention a concept that arises in the B assessment model integrated evidence. MIE refers to using model generated information such as virtual BE study result, not just to plan a pivotal study, but to serve as pivotal evidence. As you can see, models has been generating great values to cut costs and time for drug development. However, challenges and opportunities coexist. On the challenge side, specialized expertise is needed, and the expertise is not available in all firms. Novel methods usually possess complexity and flexibility, which makes it harder to be standardized, especially at their inception stage. For regulatory utility, models need to gain sufficient credibility dependent on its regulatory use. However, many times, there are a lack of data to verify and validate models for the purpose of use. Challenges always call for changes and therefore create opportunities. The strategies to counter the challenges include establishing a mechanism for model sharing, separating reviewing models from applications to make them applicable to multiple applications, and conducting targeted researches to address development need for all stakeholders. Since we have rolled out the concept of model master file last year, we have envisioned key features of model master files. First, it must have description of the model with implementation details, validation dataset, and purpose of use. Like drug master files, model master files can also be submitted to FDA separately from an application, and model master files, as mentioned earlier, submitted to FDA can be cross-referenced by multiple applications. Data in the model master file can be costly. They can be challenging to get proprietary information that have been formatted, filtered, and curated for model verification and validation against the purpose of the use. Cost and labor may have incurred because of that. They can include data that have been collected from multiple sources and related products for platform qualification. What are the potential benefits that the model master files can provide? First, it can increase industry awareness on regulatory acceptance on the utility of certain models. Second, small firms can gain access and take advantage of model master files without in-house expertise. Third, model developers as a master file owner can interact with the agency through model master file review process, once triggered by an application, on how to sufficiently verify and validate a model for the regulatory utility. Before we dive into model master files, let's take a quick look on different types of drug master files. Drug master file can assume different types. For example, type 1 for manufacturing site, facilities, operating procedures, and personnel. Type 2 for drug substance intermediate, and material used in their preparation or drug product, and etc. for other types. Here I want to mention that the Shared System Risk Evaluation and Mitigation Strategy, RAMS, a best practice in drug development, which resembles some features of modeling practice, is currently managed as Type 5 of Drug Master File. This slide shows the type of models that could be in the form of drug master files. They can include the mechanistic AVC models, PBPK models, computational fluid dynamic, and other types of mechanistic models, systems pharmacology, population PK models, explorer response analysis, PK-PD relationships. I'm going to use some examples to illustrate some potential types and utilities of model master files. They include one PBPK model for the approval of a locally acting product, one oral absorption PBPK model to justify Q3 parameter division and a safe space determination, one quantitative clinical pharmacology method for data imputation associated with dose scale analysis. The successful application of PBPK modeling for locally acting generic product approval has been shown in the case of diclofenac sodium topical gel approval. The product was approved based on a totality of evidence, including a virtual BE assessment leveraging thermal PBPK modeling and simulation instead of comparative clinical endpoint study in patients. The modeling approach demonstrated the BE between the generic and the reference product at the presumed site of action by characterizing the relationship between systemic, which is plasma, and local, which is skin and synovial fluid drug exposures. The model verification and validation process involved assessing the goodness fit for observed data of diclofenac concentrations in skin tissues and plasma, and the overall performance of the modeling platform for predicting local and systemic exposures for relevant products following the same route of delivery. This case serves as an example of using PBPK modeling and simulation for BE assessment, as well as providing an example on good practices for model verification and validation for dermatological drug products. As we can see, this application contains high level of data model platform validation, product-specific modeling parameters for diclofenac topical gel, a platform for thermal PBPK models, a practice for model building, V&V, and a simulation for a well-defined regulatory use. If the modeling portion of this application can be managed as a model master file, future applications can refer to the model master file for the same regulatory use. For the second case, PDB bronchoprovocation study included considerable amount of censored values in the PD response PC20 data. The model-based data imputation, named as the M3 model in the field of pharmacometrics, along with some sensitivity analysis, improved the credibility of the PD model and provided model integrated evidence to support the final ANDA approval as one of the first generics in 2020 as one of the pandemic rescue therapy. Technically, the likelihood-based M3 approach to perform data imputation for censored values can represent a good practice when another appropriate situation arises. In case 2, the model-based M3 imputation method can be reused by another application if it exists in the form of a model master file. Another applicant can refer to the same model master file for the same regulatory use, and the reviewer for the new application can immediately learn that this M3 method had been used and accepted for the purpose of use in the past. Therefore, this M3 method here can represent a different type of model master file, a methodology for data imputation that can gain efficiency both in drug development and the regulatory reviews. For the third case, a modeling approach has been used to assess the impact of division in particle size distribution between test and reference products. Based on PBPK model developed for this particular product that have been sufficiently verified and validated, simulations have shown that the chance for the observed D90 division to lead to bioinequivalence is low. Consequently, the PBPK modeling and simulation results supported its tentative approval. Out of this modeling practice, we have data for PBPK V&V and a specific PBPK model for drug absorption considering particle size distribution. For case 3, we see the following: a master file to use PBPK model to assess particle size effect on PK exposure, a good practice to set a B space for particle size distribution, product-specific PBPK parameters for the product involved following oral administration. All of them can be kept separately in the model master file to be referred wholly or partially when another similar situation arises. I want to give a tentative summary on different types of model master files in comparison to drug master files. Model master files can potentially be, first, modeling platform or best practices, including automation procedures and data sources to qualify the platform. Second, product-specific models and modeling parameters, including substance and formulation parameters and relevant data records. Third, computational method, including optimization routines and automation procedures if situation applies. In summary, model master files should have multiple types: increase model knowledge and information sharing, be upgradable for enhancement improvement, be reusable, make FDA review more efficient. We, FDA, along with industry and academia, can work together to define content and format model master files, to set up a process to submit, review, and manage model master files, and to roll out a mechanism to publish and announce model master files to increase the awareness of their regulatory acceptance. Finally, I want to thank Manny, who is behind the scene but contributed critically to the development of concept of model master file. I look forward for a constructive discussion in the panel after further presentations by Dr. Annie Hooker, Dr. Viera Lukacova, and Dr. Amin Rostami. Talk to you soon.
Hi everyone, my name is Andrew Hooker, and today I'm going to talk about a population PK-based model integrated bioequivalence platform. Of course, what I'm going to present here is not just me, and I've listed my co-workers here as well that have worked on this project. So the work I'm going to talk about here was actually funded by the FDA from the Generic Drugs Office. So there's 3 different contracts that we've been working on. That's the basis of this work that I'm going to be talking about here. So what are we trying to improve? Well, if we think about bioequivalence studies and we think about the standard bioequivalence study, then we have a 2x2 crossover design where typically each PK profile is defined by is evaluated by a non-compartmental analysis approach, where you then assess AUC values and half-lives and Cmax and Tmax values. And then you take a ratio of the test to reference for each individual and compute a geometric mean and its uncertainty and see whether they fall within, that uncertainty falls within the required limits. Now, there are a number of problems with standard bioequivalence studies. I think one of the largest problems is that NCA-based analyses, non-compartmental based analyses, may not always be appropriate. So there's oftentimes, or there can be, sparse data that can cause problems for NCA-based analyses. You might see drugs with long half-life, and maybe you're not able to then evaluate the drug at the end of the PK profile to get an understanding of the last phase of the PK profile. If you have highly variable drugs, then doing this summary statistic and then trying to evaluate the variability of that summary statistic is clearly not is an approach that may have more problems than looking at the data itself and modeling the data and trying to understand the variability there. You can have other problems with these bioequivalence studies and also the NCA-based approaches. So our approach in these projects have been to look at model-based methods or model-integrated methods where we can use the model to tell us something about bioequivalence. And we've seen that models compared to standard statistical methods can oftentimes give a large power increase. Here's some references for those studies that have been shown previously that model-based approaches can improve the power compared to standard statistical methods. So our approach in these projects has been to look at model integrated bioequivalence methods in the following setup. So what we've been doing is looking at taking bioequivalence data and fitting it to pharmacometrics, so population-based nonlinear mixed effects models. Those models have been adjusted so that you can estimate treatment differences on all absorption parameters in the model. And then once the model has been fit to the data, then we use that model to tell us something about bioequivalence. In the most basic setting, you could think about having a model and the model tells you a clearance value, and then you could use clearance to say, take dose divided by clearance, that would give you an AUC. But we want to do something that's not based on specific models. So what we do is a simulation setup, where we take our model, then we simulate, we take our model and first estimate parameter uncertainty based on various different methods. And within that model and parameter uncertainty, then we simulate out populations of individuals. So thousands of individuals in each simulation setting based on the uncertainty distribution of our parameters. With that simulation, then we can compute a geometric mean ratio given that, that set of parameter values and those models. And that gives us then one Cmax AUC ratio for a population. We can do that many times to get a distribution of these mean ratios, which allow us to then look at the distribution and compute confidence intervals and derive a bioequivalence conclusion. So all of this is based on a model fitting to data, and then afterwards we can use our model to tell us something about bioequivalence. And what we've seen in these studies is that we can control type 1 error. Here we're looking at a simulation study where we make some simplifying assumptions, where we assume that the model that we simulate the data with is the same model that we can analyze, or it's the same structure as the model we analyze the data with. And we can then look at simulating data with high variability or low variability, and then with various richness of data, with various numbers of individuals and various numbers of observations. And in this case, the red is the model-based approach, model-integrated approach, and the blue is the standard approach. And what you can see is that the model-based approach controls type 1 error in all of these situations that we investigated. And in the high variation situations and sparser data situations, then we actually see a dramatic increase in power for the model integrated approaches. That simulation study was based on simulating with a model and then estimating with the same model, but then adding in treatment differences in the absorption parameters. So if we don't have that situation where we know the pharmacokinetic model that describes data well in a true study, or if the model that we actually have is non-identifiable, if we're going to be running a simple crossover study, or maybe we don't have a prior model, then we need to do something more complicated. And in the work we've been looking at here, we've been looking at model averaging as a way to account for this problem. So we have multiple models that we predefine in order to evaluate the bioequivalence data. And then we use all of those models in the assessment of bioequivalence afterwards. This allows us to avoid, or at least mitigate estimation bias or overestimation of precision in our models and parameters. I've just referenced some papers that talk more about this. This is the paper that we were using, the methods that we were using, since this was something that I was involved in. And we've done then some simulation studies to investigate what happens if we don't know the true underlying model. And we can see that in this case, the The red squares is a model-based approach, and we can see that we can control the type 1 error. The blue triangles are the standard approach. And then you can see the red squares have dramatically higher power, especially in this case in Cmax, where the standard approach had only a 50% power, whereas the model-based approach had 90% power. We've also been investigating the use of models to enable us to change the types of studies that we can actually run for bioequivalence. One situation we were investigating was looking at long-acting injectables. And in many situations for long-acting injectables, you may have to give the drug to patients, and that means that you have to be giving them, you can't give just a crossover study, you have to be at steady state. So the typical study would be to have a reference drug where they're on at steady state, and then you measure the last PK profile at steady state before you give the dose of a new drug that you're testing for bioequivalence, and then eventually you wait for steady state, and then you compare the PK profile of the, once you're at steady state for the test, compared to the PK profile of the steady state at reference. With our model integrated approaches, then we were looking, or we have been looking at comparing the first profile before switching to the test product and the first profile after switching to the test product. We call this a switch study. And using models, then we can subtract out the remaining reference product from the PK profiles that we measure here to investigate then bioequivalence even with this switch study. Now, if you take a model for example, paliperidone, this is paliperidone 1 month dosing, what you can see is that when you simulate the typical individual at steady state, this is a black solid line here, and then you change, in this case, we're changing absorption rate parameters in the model to different levels. What you can see is that, in fact, the first PK profile after switching to the new compound that may have a difference in these absorption rate parameters, you can actually see that you have more difference between the PK, the reference product. So the switch study, looking at this profile, is actually more sensitive to changes in absorption rate. Whereas the switch study, if you change absorption extent in the model, so the reference product has a different relative bioavailability, then you can see that the switch study, this first profile, is not as different as what you might see at steady state. So what you can expect with these switch studies is that they will be less sensitive to absorption extent, but they'll be more sensitive to absorption rate. We then did some investigations. What happens if we look at if the product that we have is only different in absorption extent, what's the worst case scenario? And in that case, what we saw was that with these switch studies, you might see, for one example, we were looking at paliperidone 1 month, and it was taking about 600 individuals compared to 100 individuals in order to have equivalent power with these two types of studies. You can see that the model-based approach was able to control type 1 error. It was even a little conservative with the crossover steady state study, the same conservativeness that we actually saw with the standard approach. So then one can compare and see whether a dramatically shortened study with more individuals makes sense compared to a much longer study with fewer individuals in any situation where one might want to run a study. I mentioned that there was standard approaches compared to model-based approaches are oftentimes, you can oftentimes show that the model-based approaches improve power. You can also see improved power when you then optimize for those model-based approaches. And in this case, we were also able to see that if we optimize the sampling times for the model integrated approach, and even add in an extra period before switching to the test drug and measure 2 periods of reference before switching to the test, we could actually reduce the number of individuals needed by 10% in this study. So we've been looking at model-based approaches for bioequivalence analysis. We've also been looking at model-informed approaches. I haven't talked about that, where we might modify the NCA-based BE methods by changing, for example, the limits that you might use for testing bioequivalence. And when we use models to inform on those changing limits. And what we've seen is that for both of these approaches, we can reduce sample size, we can reduce study duration, and this makes bioequivalent studies more feasible and more flexible. And this is especially the case when we have challenging situations that don't work in the standard approach. Things like long-acting injectables that are used for inpatient settings, where you can't study the drug in healthy volunteers, or in ophthalmic drugs where you have very sparse data. So we think these methods make a lot of sense in situations where standard NCA-based approaches don't work, or if we want to reduce sample. So practically, how can we actually implement this model-based bioequivalence? One of the first things you have to think about is that in order to do the analysis, you need models, and you need to predefine your models. So you either need a model from a reference product, some previous model that's been published perhaps, or you need to figure out how to build a model in a predefined way that can still maintain type 1 error. Once you have your models based on some reference product, you still need to adjust those models by adding treatment sequence, period, and intercation variability effects on the absorption parameters of your model, so you can see the differences between your test and reference product. In, for example, this long-acting injectable project that we were working on, we were looking at long-acting injectables. There was 33 products already approved by the FDA. 17 of those products had published models, and there was a total of 27 published models for those 17 products. But those models were then developed on multiple studies from drug development, so they were too large to have all the parameters estimated in a bioequivalence study. So what needs to be done, even if you have a model from a publication? Well, the first thing that needs to be done is you have to make sure that all the model parameters are identifiable given the study design. This is something you might test with optimal design software before you run an experiment. You should also qualify your model by assessing whether you can simulate data that looks similar to, say, some reference data, or maybe the reference arm of the bioequivalence trial, to assess whether your model can simulate AUCs and Cmaxes that look similar to the data. So there are a number of things that need to be done, even if there is a published model, in order to make the model appropriate for model integrated bioequivalence studies. So what would be the benefit of a sort of a model sharing program? Well, this model qualification procedure could be done one time for any specific type of design or analysis, and then be shared so that other people could use that same model that's already been qualified. I think that would improve reproducibility and validation of the scientific results, so you can really see which model is being used and how it's being used. And it should allow for faster propagation of new science. If you can take what people have done in specific models and use those models in a new study. I think it's useful to be able to explicitly evaluate and verify the models that you see that are being used as well. And it will also help with model standardization. Now, where would we need a model master file, and how might that work? So I see the model master file as acting as a model repository where you can get sort of qualification for specific purposes. You can get different levels of qualification. I think there's also a discussion about a private model master file where you would not be able to see the model that you're actually using for the analysis, which I think may limit scientific discourse or innovation, perhaps something we can discuss during the discussion period. I think there's other issues with a model master file, and of course there's investment needed both in developing how this would work and maintaining that process. There's going to be IEP issues, there's going to be lots of sort of how do you get the API to work for different modeling languages and file formats so that people can actually use the information. The last thing I wanted to show was a way that we've sort of thought about this idea of giving a model to people to use for a specific purpose. So in our work, we've been developing a package called BE-mod, which includes a number of model integrated BE methods, as well as standard NCA-based methods. And you can run this in R based on a script or a Shiny platform, which I'm showing here. We've also developed a package called BE-mod LAI, and that is a long-acting injectable use of BE-mod, where we've implemented the models that we think should be used in the BE analysis for a switch study for paliperidone 1 month. And then those models are implemented and you can use them as you would like in the analysis procedure. So BE-mod will be available shortly on CRAN. We're currently getting approval from the FDA. So I think that model sharing can increase the quality of scientific work. And for model integrated bioequivalence analysis, a way to share and qualified models for specific products and situations would aid in the practical implementation of the approach. Thank you very much.
Good morning, good afternoon, good evening, everyone. First of all, I'd like to thank the organizers for inviting me to contribute to this discussion. In the next few minutes, we will talk about mechanistic IVIVC, what it is, what are the advantages, what does it take to create it. And since this is a session on model sharing and communication with the FDA, we'll also touch briefly on how it needs to be documented and reported to allow proper review by the regulators. And even though in this presentation and the example that I will be showing, I'm focusing on the orally absorbed compounds, the general principles would apply also to other administration routes. You may be concerned with different processes and with different physiological aspects for each administration route, but the general principle of why and how to do this would apply to all of them. So let's start with just a little bit of general background on IVIVC. I expect the majority of our audience is familiar with the concept, and many of you may have created some IVIVC, whether it is for internal use or for regulatory application. But just to make sure that everybody is on the same page, we'll go through a couple of slides of the basics. Within IVIVC, we are trying to find a relationship between an in vitro property and in vivo response. Most typically, this would be a relationship between the in vitro dissolution rate or dissolution profile and an in vivo response, typically represented by plasma concentration time data. Why do we do this? Helps with the dissolution method development. The process of IVIVC can guide you towards the in vitro dissolution method that best correlates with the in vivo behavior of your formulation. Once you have an IVIVC, you can use it to help develop the formulation, help to optimize the formulation, or to establish dissolution specifications or the dissolution safe space. And all of this leads to not only speeding up your formulation development, but also to reduce regulatory burden. And reducing the regulatory burden is really the major focus of this session in terms of how to report and effectively communicate the model, possibly sharing the model, to speed up the reviews by the regulatory agencies. There are several different categories or ways to generate IVIVC. Could be a single point relationships between one in vitro dissolution parameter and the PK output. Could be correlation based on statistical moment analysis, but probably the most common one, it really is the level A IVIVC, which is point-to-point relationship between in vitro dissolution and in vivo input rate. And this relationship can be linear or non-linear depending on the formulation. So what kind of data you need to generate this level A IVIVC? Of course, you need in vitro dissolution data and in vivo CP time profiles for the formulations that you are going to use to build IVIVC and validate it. Right? So this would be typically your extended release formulations. But of course, the in vivo plasma concentration time profile is dependent not only on the release of the compound in vivo, but also on the systemic disposition of the compound, how it's distributing into the tissues, how it's being eliminated, and so on. So before you get to the deconvolution step, you do need to get the baseline either PK model or the UIR, and this is typically built using either intravenous or IR data or data from immediate release formulations or solution doses to basically generate those baseline PK parameters. Once you have those, in the first step, you'll do the deconvolution. So you'll obtain your in vivo input rate for your modified release formulations. And then you find the relationship between that in vivo input rate and the in vitro dissolution that you measured in your assay. You might have noticed that for the in vivo profile, I keep referring to it as input rate rather than in vivo dissolution. And this is because depending on how you calibrate that baseline model, you might be deconvoluting different processes. So the deconvoluted profile may have different meaning. Let's take a look at what is happening with the compound after oral administration. The formulation that you swallow will be moving through the gut lumen and will be releasing the drug. The released drug then gets absorbed into the enterocytes, and the rate of this absorption may be influenced by different processes, depending on what impacts the compound absorption, whether it is passive diffusion, paracellular or transcellular, whether there are any transporters, uptake or efflux, and so on. And it can also change along the intestine. The absorbed drug may get metabolized in the enterocytes, and again, the rate of metabolism might be changing along the intestine. So only portion of the compound may actually make it into portal vein. The portal vein will carry the drug through the liver, where it can again undergo additional first pass elimination. And finally, the rest of the compound makes it into the systemic circulation, which you eventually measure in the form of plasma concentration time profile. And depending on what data you used to parameterize that PK, you might be, or what model you are using for the deconvolution, your deconvoluted profile might be anything from the in vivo dissolution all the way up to simply deconvoluting the rate of the drug appearing in systemic circulation. In the simplest case, let's say if you simply used in vivo CP time profiles to deconvolute the in vivo input rate, you are basically lumping everything that was happening between the dissolution and appearing in the systemic circulation into that single rate or the single profile of compound appearing in systemic circulation. And for a good number of drugs and formulations, this works nicely. If there is no nonlinearity or any process that would change the time dependency of these deconvoluted profiles, you might be able to find a good and predictive relationship between this rate of drug appearing in systemic circulation and the in vitro dissolution rate. But as you've seen, there is a number of processes that we covered by this gray box, which can throw a wrinkle into that whole process and prevent generating that simple IVIVC. Some of those most obvious processes are highlighted on this slide. As you would expect, they relate to expression levels or to activities of uptake, efflux transporters, or the enzymes throughout the intestine, right? And all of these have, or most of these have non-uniform distribution throughout the intestine. So typically they would have higher expression in certain regions of the intestine, lower expressions in other regions of the intestine. So this could impact the rate of the systemic of the compound showing up in the systemic circulation different than what is the actual rate of the compound in vivo. So the differences in the rates in the systemic circulation could be magnified compared to what is the actual difference in the in vivo dissolution rate for the two different formulations, depending on what the distribution of the transporter is along the intestine, what the distribution of the enzyme is along the intestine, or other possible processes, whether saturation may be playing a role, and so on. So with all of these, if this starts playing a role, you would have trouble building that IVIVC. That is exactly what the mechanistic IVIVC is meant for, is to parameterize a model which accounts for each of those processes, which accounts for the actual mechanism of drug being absorbed into the intestine, and how the how the transporter, whether it is uptake or reflex transporter, is distributed along the intestine. Account for possible first pass, and again, how the enzyme that would be metabolizing the compound is expressed and how its activity is changing along the intestine. Account for possible saturation of that first pass extraction, whether it is in gut or liver, and so on. So when you have that full model and use the full model accounting for all of these processes to do the deconvolution in vivo, now you are really deconvoluting the release of the compound in vivo in the gut lumen, and everything else is covered by the model. So to calibrate such a model, of course, you do need more data. Ideally, you would be using IV data to parameterize the systemic disposition, some immediate release oral absorption data or oral data to calibrate the subsequent processes, the absorption, first pass, and so on, and then use that full model for deconvolution. Just one quick example. This is a fairly old example published about 20 years ago for metformin, where this group tried to create an IVIVC. They had 3 nice formulations with significant differences in the in vitro dissolution as well as in vivo exposure. There are 4 profiles here for the in vivo exposure because the circles, the first one is actually the IR solution, which was calibrated or used to calibrate the input function, and the remaining 3 are the formulations corresponding to these in vitro release profiles. And trying to build a simple IVIVC was not very successful because in addition to changing the rate of the release in vivo, the formulations or the extent of the absorption, the fraction absorbed, the total fraction absorbed was also changing for the formulations. So for the equation that would be nicely matching the fastest releasing formulation, they would be over-predicting the exposure for the slower releasing formulations. And in order to get around that problem, they had to actually create additional relationship. So not only relating the in vitro release profile to in vivo release profile or to in vivo absorption profile, but also relate the in vitro release profile to the extent of the in vivo absorption and create additional empirical relationship to scale the maximum fraction absorbed related to the rate of dissolution. And that would then allow them to create a nice predictive IVIVC. This is something that could be explained by the mechanisms for the metformin absorption. And there might be still some discussion about the exact mechanisms. One of them was highlighted in this study about 15 years ago, where the authors proposed that the actual effective absorption or the pathway by which the metformin actually makes it into the portal vein and systemic circulation is only the paracellular pathway. But it is also a substrate for bidirectional transporter on the apical side of the enterocytes, no transporter on the basolateral side, But this apical transporter on the basolateral, on the apical side, the bidirectional transporter, would first cause the uptake of the compound into the enterocytes. And as the compound in the gut lumen moves towards the lower regions, the concentration gradient changes, then it would start moving the compound back into the enterocyte, into the gut lumen. So basically, this recycling through enterocytes creates sort of a temporary depot of the compound and allows for the compound to be absorbed at the later times as well. The problem here is that the paracellular pathway in human is most efficient in the upper small intestine. So the formulation that releases the compound quickly provides the compound available for the storage in the enterocytes in the upper small intestine. And as that gets released back into the gut lumen, it can still get absorbed into the systemic circulation. The formulation that does not release the compound until it reaches the lower small intestine or colon could still recycle the compound through the enterocytes. But the problem is that the paracellular pathway is not there anymore, so as the compound moves back into the gut lumen, it really doesn't have a way to get absorbed and it just gets excreted. So that would explain why the slower releasing formulation resulted not only in slower rate of appearing in systemic circulation, but also less complete extent of the absorption. And this is something that was presented. It wasn't published in a full paper. It was just the preliminary model that was presented at the AAPS meeting about 8 years ago or 7 years ago, where this model, the mechanistic model, was introduced or was built to account for these mechanisms. So incorporate the absorption by a paracellular pathway, incorporating the impact of the transporters. And you can see it wasn't still perfect. There were still some mispredictions on some of these formulations, but it was a lot closer than the models, than more simpler models, whether it was a simple Luriegelman or even numerical deconvolution, which did not incorporate that additional empirical scaling for extent of the absorption. Right? That additional empirical scaling was really required to get the good predictions. In addition to that, the mechanistic model actually provided the explanation as to why that empirical scaling would be required with the simpler method. Right? The model did predict that if the compound doesn't get released quickly enough, it just won't have a pathway for being absorbed, and it was able to explain why that additional scaling would be needed. So what does it take to build mechanistic IVIVC? Of course, the first and foremost, determine what critical mechanisms would be impacting the drug absorption in pharmacokinetics, which mechanisms might be changing that relationship between the in vitro and in vivo release profile, and which mechanisms do need to be incorporated in the model. Then it comes to parameterizing the model. Ideally, you would be using intravenous data to parameterize the systemic disposition model, immediate or data from immediate release formulations, whether it is solutions or tablets, to parameterize the absorption of the model, to have a complete model accounting for all the mechanisms impacting the compound everywhere from the absorption until appearing in systemic circulation. Then you would use that model to deconvolute in vivo release profiles and finding relationship between the in vivo and in vitro release profiles. So these last two steps are exactly the same for any type of IVIVC, but the first step is really the most critical one for building that mechanistic absorption model. And this is where using a PBPK model is a really great tool for that type of development, because this model, or the PBPK model, is really a system of differential equations that combine the compound-specific properties like solubility, permeability, Km and Vmax values for interactions with the transporters, enzymes, and so on, the formulation parameters, so how the formulation is moving through the intestine, where and how it might be releasing the compound, and finally the system or physiology-specific parameters. Again, this is something that would be impacting the transit times through the intestine, what the enzyme and transporter expression levels would actually be in the intestine, how they are changing throughout the intestine, how the environment that would be impacting the release of the compound or the dissolution of the compound would be changing along the intestine, and so on. Right? So these are provided, these are the physiological parameters, and would basically be staying constant, are not impacted by the formulation of the compound and could be reused for different compounds and formulations. So with all of these parts, to build the mechanistic IV-IVC, what do you need to actually provide to the regulators to be able to efficiently review or evaluate that application? Right? One is describing the model structure, which processes are affecting the drug absorption, which processes are affecting the systemic disposition, how they were incorporated in the model, why they are critical. Right? And this is something that would be provided by the sponsor, but may be available from the public sources. As different groups are doing research on the same compound, this information might be coming up. The next one are drug inputs. Physicochemical properties, block P, Km, Vmax values, pKa values, and so on. And again, this should be provided by the sponsors, but may be available from a variety of public sources. The system inputs, this may be the simplest process or the simplest way of reporting, especially if you are using some of the established or more commercial PBPK platforms, because all of the physiological parameters are already included in those. Right? These are the parameters that are independent of drug or formulation, come with the PBPK platform, but in order to make it clear for the reviewer to actually evaluate, the sponsor needs to clearly define what platform was used, what platform version was used. Right? And if they modified any default settings, they will need to provide justification, explain which default settings were changed and why they were changed, because that would now create a difference from any validation that was done previously, and they might need to revalidate those default settings. And finally, for IV-IVC application, specifically for IV-IVC application, you of course need to provide the additional information in terms of the in vitro dissolution data, what IV-IVC equation you ended up with, what is the type of in vitro dissolution assay that IV-IVC equation would apply to, and so on. And this is something that really needs to be provided by the sponsor because it is specific to that one IV-IVC that was generated. So to sum this up, the mechanistic IV-IVC provides IV-IVC utility to compounds undergoing complex mechanisms. Creating successful mechanistic IV-IVC requires building a complete mechanistic absorption model. The negative of that is it is much more involved process than simply fitting compartmental PK to IV or IR solution data. But the effort pays off because it does provide you with additional information of what impacts the exposure. And for certain compounds, for certain applications, it may really be the only way to actually build an IV-IVC. And the description or reporting of the model does require reporting the model structure, compound-specific information, system-specific information, and the formulation, which would be specific for the IV-IVC applications. And some of these could be potentially reusable as they are available in the public sources. Some of these are coming with the platform, especially if you are using some of the more commercial or provided supported platform. The information is already in there. You don't have to worry about validating it. But there are some pieces of information that really have to come from the sponsor because they are really specific to that one application that was provided. And I think I will stop here and we can discuss during the panel discussion any questions on this presentation.
Hello, everyone. It's a distinct pleasure for me to be able to contribute to the discussions we are having over the model master file as part of this particular workshop. Before I start to talk about the model reusability, which is the main topic of my talk here, I would like to take everyone through the journey that we have had over the last 20 years in relation to incorporation of modeling and simulation into practices involving drug development through the life cycle of the any medicine that comes to the market. This is something that I have coined as starting from modeling and simulation in the form of toys for big boys, which was very limited corner of what modeling and simulation could offer, to come to a point that modeling is now done by all for all. So it involves population level modeling and also much bigger community of the modelers. I'm going to use my own 4 stops of the journey that I had and go through the stops that they are signified by 4 papers that I'm going to bring up, analyze, and discuss with you. This is our stop 1. This paper was published in 2012 in the journal Clinical Pharmacology and Therapeutics, and the bottom left-hand side corner is the key element of what this report was talking about as part of my own journey, as I explained to you, and that was the importance of separating systems parameters from the drug parameters and putting them back together under different conditions, different populations that trial design then governs in any virtualization of that particular clinical trial. And I intentionally in that particular report talked about the offerings that such modeling can have with regard to the very dynamic system of oral drug absorption. Even this was not one of the, I would say, that very popular areas of application of the PBPK at the time. The slide here indicates word cloud analysis for that particular paper because I wanted to, for myself, to understand what was specific about that paper, which many people now as considered as one of the key papers, and it has been cited many times, apart from the model and drug. And while you can see that there are many words such as data, enzymes, IVIV, PBPK, but the third biggest emphasis apart from the drug and model was the systems. And we will come back to this later on and its importance with regard to reuse of the models and keeping the system parameters same for the target population once they are verified. Now, fast forward to 2018 with another paper in Clinical Pharmacology and Therapeutics, when I was asked to talk about the value of reverse translation and use of observed clinical data together with the PBPK and bottom-up approach. In reality, we had already published in 2014 another piece together with my colleague, Professor Leon Arons, and one of our PhD students at the time, Nikos Samandouras, in the British Journal of Clinical Pharmacology, where we argued that we should not put bottom-up modeling against the top-down, but we should be actually combine those. And that's the reason for the first time we basically put bottom-up and top-down approaches, as opposed to what people were debating at the time and putting top-down versus bottom-up modeling. And we will come again to the discussions on model master file and the importance of this particular stage. And the word cloud analysis shows that apart from, again, model and system data, etc., the other element here were the parameters, recognizing the parameters that they apply, and not all the time we are able for mechanistic models to get all of them from in vitro setting, and hence when we are combining these models, we have got the advantage of getting system parameters and interactions with the drug, and yet not to be confined to what classical PK models offer with regard to inability to extend beyond the set of data that they are investigating later on. Fast forward to year 2021, we published another piece that was related to the survey our team had done on applications of the PBPK models and comparing it with the classical PK growth over the last 20 years. As you can see from the graphics in the right-hand side, it was immediately obvious that the PBPK growth more than 40-fold over that period of 20 years was much higher than what was happening with classical PK, with a growth of around 4-fold, aligned with the general increase in the size of the scientific publications. This helped us to start in the debate with some other colleagues who were questioning the value of the closed source modeling against the open source code modeling, which was the common way of modeling by writing your own code and every equation and so on. And we argued that while we are not challenging many of the benefits that the open source code modeling offers, particularly within the academic community, however, once these models are established and they are qualified, wider usage of closed systems models when the application is the main subject area is going to be much easier, and it attracts more modelers. And all the data that they were showing, they were in favor of that particular argument. This has implications with regard to the trust that people put on any scientific work, and this is not just specific to the modeling, because reproducibility of any piece of work is very important, regardless of the regulatory submissions, which are even more important, that you have got reliability on what is happening with such models. And one of the problems that these open source models, whether we like it or not, they have been susceptible to, they have not been able to satisfy some of the criteria for the reproducibility, because the control over them and ability to keep an eye on different changes that they are happening and so on, they have not been as, I will say, robust as the closed form systems. And the analysis of this particular paper station that, in our journey that we had, obviously puts the platform and particularly the commercial nature of some of them into the spotlight, and indicates that these have not been against modeling, but they have actually helped with the expansion of the modeling. Of course, the quality issue, when it comes, whether it is open source code or whether it is closed systems, is the paramount factor that we should consider. And there are many different words that they are used in this area. Validation of the code and its operation with algorithm is one of the things that is possible, but unfortunately the term, in my view, is used loosely with regard to the overall validation of the aim and target of the modeling, even when it is applied to an area that we know that we are not going to have clinical data to show what people call validation. And hence I tried to separate the validation of the code from verification of the platform in that particular area and its qualification on certain applications as an indication of model credibility to go and state something about an area of research and some clinical angle that we do not have actual any data. And of course, here we have got many different views with regard to the number of cases that we have to provide for the verification and so on. But the most important discussion is the separation between the verification of the platform and system parameters and so on versus the model verification for the particular specific area of the usage that we are talking about, and these are explained in the paper. Our fourth step is this unpublished piece of work that we have got under review. And I will go slightly, I will say slower on this because obviously they are not available yet. And that is explaining how when we follow up any original PBPK work, this is the top layer of the two segments that you are seeing above and below the dashed line. For the open source code models, which are the two examples in the right hand side, and the closed source code models that you are seeing in the left hand side. When we look at the reusability of overall, we looked at 145 cases. In each of those pie charts above the dashed lines, you will see the blue area is indicating what proportion of all those models developed with that particular platform and the non-open source code versus open source code modality were used later on. And you can see the blue area in the case of closed source code systems looks higher than the open source code examples. However, that's not hugely different. But when you look at the bottom pie charts that they are coming from those blue areas, particularly the pairs that they are the one side of the pair that is in the right hand side, when you look at the external use, this is basically usage or reuse of that particular model by people who had no links with the original developers. You can see the proportions in the case of the non-open source code modality is much higher, and majority of the reuse of the open source code are what we call internal, which basically means somebody from the original team who developed that was using it. And this is very important. These are some of the definitions that we actually borrowed from the software side versus modeling. We did not find any useful definitions for the reusability in the area of modeling. Therefore, we went to the software area and modified what they are using in those areas, and my slides are available and you can study these later on. But the bottom line is the star analysis that you are seeing in the left hand side, and that is saying the likelihood of a non-open source code model to be reused by people who were not party to the development of the original one is twice more than the reusability of the systems that they are generated with open source code modality. Therefore, now how this is going to impact actually model master file. This is basically saying that the quality that is required to go into any reusability, model master file is basically built on the back of reusability under different conditions with a new drug, with a new formulation, and so on. When you are changing the process of the development that you have got for formulation and so on. So the quality element is much easier to not only to keep and have robustness with regard to the quality, but also assessment gets easier. because you don't have to go through every single line of the code, particularly knowing that these are multi-layer systems with many, many lines of coding and the values that they have actually gone through the system elements of those models. But there are still, I would say, open questions that people need to discuss, and that is for the closed systems. Of course, not everybody has got access to the code, so how comprehensive the models are, and if we need to change some of them, they need to be done by certain individuals who have got the let's say they are key holders for that, and who they are if it is open source code. Because at the moment with open source code, everybody can do that. With the closed systems, how we ensure that the frequency of these certifying and redoing some of the models are high enough to cater for the latest knowledge, number of the required verification cases for areas, what actually is acceptable, and obviously, the transparency. When people talk about that closed system, and we have argued that there is a difference between open science versus open source code. If we are claiming that these are not black boxes, and I have put in the past as glass boxes, they are glass boxes, but you have to have tickets to go and actually meet them in a sort of a museum environment or whatever. You can't, not only you can't poke into it, but you may not be allowed. If you are actually in a competitive environment, why you are supposed to be allowing other people to come and re-engineer what you have done? But for a regulatory perspective, of course, you have to actually be able to show how they are working. Now, this changes the philosophy, particularly in the pharmaceutics area, because this is basically putting what we typically do with the systems as biopredictive dissolution or any other in vitro system under question, because it is biopredictive in what condition, what sense, and how do we know that particular system under different condition also will perform, or we have to do it under different conditions. But instead, it is saying that we have to open up these in the process that basically it is described in this particular chapter that we had, with again modeling of the in vitro data, and in the same way that we were doing in the DMPK area, by analyzing and getting the values that they are more fundamental and intrinsic, to get the formulation information in the same format and then plugging them back into these quantitative models. In the process, you will be able then to cater for more than the target population that originally you were catering for. And this is, sort of going beyond what people call these days fit for purpose, which I have got issues with, because I call it fit for current purpose, but framed for future questions that you may have, is what we should be doing. We have to leave them open and flexible. So framed for future is something that we have to always think of the models that we are doing. And this is important on the basis of examples that we have recently put on the virtual bioequivalence, with the within subject variability being a key issue for that. And of course, when we get these values, we need to incorporate them into a system after we have verified them. One of the verifications that it's not that easy to go and just from the literature and pick up the within subject variability either for PK parameters or even better for the physiological parameters, because they are going to propagate and come to the PK. The reason they are not easy and therefore it takes a long time to get these system parameters are the fact that we are not repeatedly sampling from individuals, and therefore if all of us we have got the same mean value, but each of us within ourselves we have got some variability, if we pick up only one sample, we will end up with a variability which has nothing to do between subject variability, but everything to do with the within subject variability. Therefore, the current lack of repeated sampling, it basically gives us a mixture that is fusion between subject variability and within subject variability. But doing this work, progressing it, and incorporating it into a system analysis, a model master file, requires doing analysis for different sets of drugs and formulations, something that we haven't done yet. We have talked about it, but we haven't done it. But we have shown that how these may impact the extraction of within subject variability that explains our observations. And this is like many other fitting that we used to do for the concentration time profile. It basically says that the model is not identifiable, but to make it identifiable, we need sets of different drugs and different formulations to capture those. Now, if these are gathered and verified, why do you want them to repeat every time? You want just to incorporate them into systems that they can be reused again. And I stop here and will be delighted to answer any of the questions during the discussion later on. Thank you.